Hello friends, today's our topic is pure culture preservation methods. As we all know, in a microbiology laboratory, maintenance of pure culture requires frequent attention so that the microorganisms are neither lost nor contaminated. For this, we have to repeatedly subculture the organisms onto suitable media. But repeated subculturing has got several disadvantages. It may lead to some mutational changes in the strains of organism so that they may lose some of their cultural, morphological or immunological characteristics. Then, how can we maintain the pure culture of microorganisms in the laboratory for future use? There comes the importance of pure culture preservation techniques. First, let us see why do we need culture preservation methods. In a microbiology lab, as we all know, we are constantly in need of pure culture of different microorganisms for different laboratory sessions, research work, and for other particular procedures. We usually call them as the stock culture. Pure culture of all microorganisms also have to be retained as reference strains for future taxonomic studies. All the microorganisms that have been identified till date are retained as reference strains in different central collection units. American Type Culture Collection or ATCC located in USA and Microbial Type Culture Collection and Gene Bank MTCC located in Chandigarh of India are such central collection units. In these central collection units, millions of microorganisms are preserved in ampules as reference strains. Now let's see the objectives of culture preservation. Culture preservation is the maintenance of microorganisms for extended period of time in viable condition without any contamination. Organisms also should not lose their genetic and morphological characteristics. Several preservation methods are available nowadays, but no method is applicable for all types of microorganisms. Preservation methods either will stop or slow down the metabolic activities and multiplication of microorganisms. Based on the period of storage, we can divide the culture preservation methods into two types, that is short-term preservation methods and long-term preservation methods. Agar slant cultures, which is otherwise known as subculturing, and refrigeration is the most common type of short-term preservation methods. There are five different types of long-term preservation methods, which includes mineral oil or liquid paraffin method, saline suspension storage, drying in vacuum, storage at low temperatures, which is otherwise called cryopreservation, and lyophilization or freeze drying. Coming to the first short-term culture preservation method, subculturing and refrigeration is the most common type of culture preservation method that is practiced in most of the microbiology laboratories. Here, we will be periodically subculturing the organisms onto suitable culture media, and we called it as the stock cultures. The selection of media, the storage temperature, and interval between the subculturing will depend upon the type of organism we are cultivating. Usually, solid media is preferred over liquid media for subculturing and refrigeration because during the growth, microorganisms will be producing waste products. In solid media, these waste products will diffuse into the slant so that it is away from the organism and it will not affect the growth of the organism. If it is liquid media, these waste products will affect the growth of microorganisms. The most common type of media used for this purpose is nutrient agar. Most of the heterotrophs can be easily maintained on nutrient agar. In this method, nutrient agar is prepared in screw-capped bottles so that the drying can be prevented. But disadvantage of this method is that repeated subculturing is needed, which may lead to mutational changes in the organisms. In this method, the nutrient agar slants prepared are inoculated with the organisms and they are incubated at a suitable temperature for 24 to 48 hours to get sufficient growth. Once we get the sufficient growth, we can transfer it into a cool, dry place. Usually, it is to the refrigerator. The temperature inside the refrigerator is 4 degrees Celsius, which will slow down the growth and metabolism of microorganisms, which will result in less nutritional use by the microorganisms. So, in that media, we can retain the organisms for longer periods of time. However, as we already mentioned, periodic subculturing is necessary for this method, which we mentioned as a disadvantage. Coming to the long-term preservation methods. Mineral oil or liquid paraffin method is a common method used for long-term preservation. Here, agar slants in screw-capped tubes are inoculated and incubated till good growth is obtained. Then it is covered with sterile mineral oil or liquid paraffin to a depth of 1 centimeter above the slant. This method can be used to preserve bacteria and fungi in viable condition at room temperature from months to years. Paraffin layer prevents the dehydration of the media and also produce anaerobic condition so that microorganisms remain in a dormant state. This method has got several advantages. The unique advantage of the method is that subculturing can be done without affecting the stock culture. In this method, subculturing is done by taking loop full of growth under the oil from these slants, then touching the loop to the glass surface of tube to drain off excess oil and then inoculating it to the fresh media. It is found that many species remain viable for longer periods under the oil than in tubes without the oil. This method is useful mainly for the strains that are susceptible to mutation on repeated subculture. This method is also considered as a simple and economical method as the equipments like desiccator, vacuum pump, etc. are not needed to carry out this procedure. The next long-term preservation method is saline suspension storage. This is a very simple and economical method. We all know sodium chloride in high concentration is inhibitory to the microorganisms. So in this method, we are making use of a sublethal concentration of sodium chloride. That is 1% of the salt solution is prepared here for the preservation of culture. Here, the bacteria is suspended in the salt solution and we have to make use of screw-capped tubes in order to avoid evaporation. Then these tubes are kept at room temperature and we can subculture onto the suitable media as and when required. This is a very easy method for storing bacterial culture for 2 to 3 years. And this is very useful in the laboratories where limited equipment facility is available. Coming to the next method of long-term preservation, that is drying in vacuum. Organisms are found to survive longer if they are dried in a desiccator than in air. Different methods are employed for drying in vacuum. For performing drying in vacuum, we require two things. One is a desiccator and other one is a vacuum pump. Here we can see the diagram of a desiccator which can be connected to a vacuum pump. The desiccator has got a lower chamber in which usually we keep the dehydrating agent. And above this chamber there is a perforated plate. And on this plate the microorganisms in suitable containers are kept for drying. A common method of drying is known as liquid drying or L drying. In this method, the microorganisms are suspended in a liquid media and then it is kept inside the desiccator over the perforated plate. It will be dried using the dehydrating agents such as sulfuric acid or phosphorus pentoxide. This method is very much suitable for the microorganisms which are sensitive to freezing or lyophilization. In this method, the freezing of the liquid media is not done as vacuum is involved. Instead of liquid media, we can also make use of sterile cover slips, filter paper, small test tubes or ampules for drying the microorganisms in the desiccator. Coming to the next long-term preservation method, that is freezing and cryopreservation. Freezing at very low temperatures is usually known as cryopreservation. Glycerol suspension method and liquid nitrogen method are the most commonly used cryopreservation techniques. In glycerol suspension method, the organisms are suspended in nutrient broth containing 50% of glycerol. Then it is frozen and stored at minus 29 degrees Celsius. We can maintain the cultures up to 2 years by this method. The availability of liquid nitrogen provided another useful means for preserving stock culture. In this method, dense cell suspensions are prepared in the medium containing cryopreservative agents like glycerol or dimethyl sulfoxide, which is commonly called DMSO. These agents will prevent the ice crystal formation during the freezing process, which may damage the bacterial cells. After cultivating the microorganisms along with the cryopreservating agents, the suspension of microorganisms are transferred to small ampules or vials and they are frozen at controlled rate to minus 150 degrees Celsius. The ice crystal formation will not take place as the cryopreservative agents like glycerol or DMSO is present in the suspension. Then these ampules or vials are stored in a liquid nitrogen refrigerator either by immersion in liquid nitrogen, that is at minus 196 degree Celsius, or it can be kept over the liquid nitrogen in the gaseous phase, that is at minus 150 degree Celsius. The liquid nitrogen is in the liquid state when it is at minus 196 degrees Celsius and it will move to a gas phase when it is at minus 150 degrees Celsius. Both these methods are employed in liquid nitrogen refrigerators. This method is also very useful for the preservation of microorganisms that cannot withstand the freeze drying. This is a very effective method and by this method cultures can be maintained for 10 to 30 years and without any change in their cultural or other biological characteristics. But the disadvantage of this method is that it is a very expensive method as liquid nitrogen has to be replenished at regular intervals to replace the loss due to evaporation. This method or lyophilization, which is otherwise known as freeze drying, is not suitable for all types of microorganisms. It can be used for only organisms that can survive the freezing. This method is mainly used to preserve the organisms that would be killed by the ordinary drying process. In this method, thick suspension of microbial cultures are prepared in small vials and then frozen to minus 60 to minus 78 degree Celsius. Then these vials are connected to the high vacuum of the lyophilizer so that the ice present in frozen culture will undergo sublimation. We all know that sublimation is the evaporation from solid to the gaseous stage without passing through the liquid phase. This will lead to the dehydration of culture media as well as the dehydration of bacteria with minimal damage to the delicate cell structure. Thus, the bacterial suspension will become a powder form. The vials containing the lyophilized organisms are sealed under vacuum and then stored in the refrigerator. This method is very effective in maintaining different types of bacteria, and culture can be maintained in the viable condition without any change in its characteristics to more than 30 years. As the cultures are preserved in this method in small ampules, only minimal storage place is required for maintaining these cultures. Also, it is very convenient to mail these vials in sealed containers to other laboratories. The lyophilized cultures have to be revived before using. First, we have to rehydrate the lyophilized vials by adding liquid medium into it, and then it has to be transferred into the soluble media and you have to incubate it. In this slide we can see the image of a lyophilizer. Here we can see the ampules connected to the high vacuum of lyophilizer. Second image is of the lyophilized cultures of microorganisms. So here we conclude our topic on pure culture preservation methods. Thanks for watching. See you soon with another topic. Bye.